All right, welcome to part two of the video lecture for chapter three. You'll recall that toward the end of the first part of the video lecture for chapter three, we had talked about the first pattern of evolutionary change, which was that modern characters typically evolve from pre-existing characters. There are about a dozen of these patterns. That was the first one. And remember that at the very end of that video lecture, with regard to that first pattern, where modern characters evolve from pre-existing characters, we got into a discussion of homology or homologous structures. Remember, these are structures or features in modern species that have been inherited from equivalent features in their ancestors. And if we have truly homologous structures, then they tell us very important information about ancestry and evolutionary relatedness of species. All right, remember also that we talked about how homology is identified. So we have correspondence of position. Where in relation to other structures is a character found? That can be very helpful. Correspondence of structure. What parts is the structure composed of? That can tell us a lot about whether or not the structure is homologous in two different species. And then finally, when mapped on an evolutionary tree, does the distribution of that feature suggests it's been inherited from a common ancestor? That can be very insightful as well. All right, an important point about homology is that it's possible to have features in two organisms that suggest common ancestry, that suggest that the feature is actually homologous when in fact it's not. When that happens, what we have is a process called homoplasy. Okay, homoplasy. Homoplasy results from various evolutionary processes. The first one is called convergence or convergent evolution. And this is where similar characters evolve from different developmental pathways, all right? It's very possible that you can have organisms on opposite sides of the planet living in very similar environments, like deserts, for example. Think about the Atacama Desert in Chile, for example, in South America, and compare that maybe to the Kalahari Desert in Southern Africa. It might be very similar environments. You can imagine also that because of that, they might present very similar selection pressures to the organisms that live in them. In other words, organisms that live in two different deserts largely may have to deal with the same basic problems. Very arid environment, maybe a very hot environment, that sort of thing, lack of food. And so evolution might come up with the same basic sets of solutions in two widely separated yet very similar environments. And so it's possible for two organisms living on different sides of the planet to evolve extremely similar adaptations to deal with the same sets of problems. If you see those features in two organisms, one from South America and one from Africa, you might suspect it's a homoplasy. You might suspect it's homologous, suggesting that they got it from a common ancestor when maybe they didn't at all. Maybe it represents two separate evolutionary innovations of the same basic solution to the same basic problem. And in that case, that feature, whatever it is, would be a homoplasy, suggesting relatedness where there is none. Good example of that. This is an example of homoplasy from what's called evolutionary convergence. Here on the left, you see the typical vertebrate eye, right? Think about a human eye, for example. You see the lens, you see the pupil, the retina on the back with the optic nerve attached. On the right, what you have is the octopus eye. So this is what you find in octopuses, or octopi rather, squids, that kind of thing. Overall, it looks very similar. You have a lens, you have a pupil, you have a cornea, there's a retina on the back with an optic nerve or optic ganglion attached. But what we start to see if we look at this a little further is that these are actually designed quite a bit differently. If you look at the vertebrate eye, you can see that the optic nerve runs in to the back, and the nerve fibers actually run over the surface of the retina and converge at the optic nerve and create what's called a blind spot. We all have that. That is not what happens in the retina of the octopus. the optic nerve or the ganglia come in and attach to the back of the cells in the retina. And so there is no blind spot. But overall, you can see that it's very similar design. There are only so many ways that a biological system like this or structure like this can collect light, focus it, pass it through the vitreous humor, which is the space here, and focus it on a retina and convert that light signal into electrical impulses that can be interpreted by neural tissue in a brain. Basically what you see here is the best way to do that, and it's evolved different times independently. One major way of doing it in the cephalopods, invertebrates, and another major way of doing it in the vertebrates. And eyes of very complex design like this, or very rudimentary design even, have evolved hundreds of times independently throughout the history of animal evolution on this planet. All right, this is a good example of convergence. You have the same problem that these two different groups of organisms are dealing with. How do you focus light and be able to use light to sense your environment? And you see that the two different groups of organisms, animals here, have converged on the same basic solution. But there is no common ancestor to both vertebrates and cephalopod invertebrates that had that structure. So this structure you see here is not a homology. It's homoplasy due to evolutionary convergence. All right. There's another way that we can get homoplasy, and that's through what's called evolutionary reversal. In reversal, you get a return to a previous ancestral condition or state, and the whales are a great example of this. Whales, so modern whales, which we know are mammals, secondarily evolved a fish-like ancestral body form from a terrestrial four-legged mammal, the derived form. Mammals evolved on land. There's no doubt about that. The fossil record is very clear. But in the case of whales, we have a group of mammals, one lineage of mammals, that secondarily moved back into the watery environment, okay, the marine environment. And so what you're seeing here is the fossil record, such as it is, for this group or this lineage of mammals. You have Indohyus, which was a species that probably lived about, say 50 million years ago, something like that. Various features of its anatomy suggest that it could have been potentially semi-aquatic, maybe like modern hippos are, for example. Further on in time, we have Ambulocetus, which would have lived in shallow waters and used lakes for swimming. You can tell that based on the structure of the legs, structure of the digits on the foot and the hand, basically. And also, these guys have some ankle bones that are extremely similar to what we find in modern artiodactyls. Artiodactyls are the hooved mammals. We actually think whales evolved from a group of hooved mammals. Okay, hooved mammals are things like the ungulates today. Okay, Rhodocetus, a little bit further on in time, definitely taking on more of an aquatic body form. You can see becoming elongated, having feet that are sort of jutting backwards, very elongated digits for helping to swim. And then finally, you see we have Durodon, and then this one here. So this is essentially a modern harbor porpoise, and you can see how Durodon, which is extinct, looked a lot like it. In fact, right there you see vestigial pelvis and hind limb bones that were just embedded in tissue. And although modern porpoises don't have that, modern whales do. If you ever go to a museum and see a skeleton of a modern whale hanging from the ceiling or whatever, look for it. You'll see these bones suspended in midair, usually by a wire or something, indicating that in life they were just embedded in tissue. They're not used, but they are the remnants of these pelvic bones that you see here. This is a case of evolutionary reversal. So if you're looking at if you know nothing about life on planet Earth and you're looking at a killer whale, a shark, and a human, and you're asked which two are most closely related, you're probably going to say the shark and the whale because they look so similar. Actually, the correct answer is the whale and the human. The whale and the human are more similar because they're both mammals. The whale and the shark, however, look very similar because that fishy, fusiform or torpedo-shaped body is what you need to move through a very dense environment, dense fluid like water. And so that's an example of evolutionary reversal. It looks like the whale and shark are more closely related when actually they're not. That's homoplasy. All right, so another example of this, of species sort of evolving the same basic solutions to common problems in their environments that might be widely separated by distance. Another good example of that is convergent evolution in nectar-feeding birds. What you see over here on the left, these are two species of honey creepers. This is a South American species. This is a Hawaiian species. Over here on the right, you see a hummingbird from Costa Rica, and down here you see a songbird from Africa. But all of these species are specialized to feed on nectar from flowers, and so they all have that sort of long, recurved beak like you would typically see in hummingbirds. But there's no common ancestor to any of these that has that beak structure. It's an example of something that in this case has evolved at least 4 separate times. And there are lots of other species of birds that have this basic structure as well, because that's the tool you need if you're going to feed on nectar in very deep potted flowers like that. All right, so just another example of homoplasy, again, suggesting relatedness where really there is none. All right, another result of evolutionary convergence and that sort of thing, in other words, homoplasy, is what's known as mimicry. Mimicry is a result of convergence, and this is when you have species that are resembling each other because one or both of them gain some sort of benefit from that. There are 2 basic types of this. First we have is what's called Batesian. This is where you have a palatable or a non-toxic or non-noxious species that comes to look like a toxic or unpalatable species. This is actually very common in a lot of insects, especially butterflies and flies. Okay, so here, for example, you have what looks like bees, right? These are actually not bees at all. This looks more like a wasp, but these are actually all flies. That's a fly that looks like a wasp. These are 2 flies that look like the big bumblebees that we see. And so if you're a predator and you're feeding on insects, you're probably going to avoid these guys if you've ever tried to catch a bee or a wasp and been stung by it, even though these are not wasps and bees at all. They're flies and they cannot sting. So these are species of flies that are mimicking either wasps or bumblebees, and this is a great example of Batesian mimicry. All right, here we have another example, perhaps the classic example of this. The monarch butterfly is known for being toxic. So the caterpillars of monarchs feed on milkweed plants, and those milkweed plants have compounds called cardiac glycosides. And these build up in the tissues of the caterpillar, the larval form, and when it metamorphoses into the adult monarch, those cardiac glycosides remain in the tissue. If a bird comes along and eats a monarch, it's going to spend a good amount of time after that violently vomiting that monarch back up. So if that happens to it, you can probably imagine that if it comes across a viceroy, which is not a monarch at all, different species, it's probably going to avoid it because the viceroy looks a lot like the monarch. But the viceroy does not have those cardiac glycosides. It's not noxious at all. The bird could eat it just fine, but the bird's probably not going to eat it because of the experience it had with the monarch before. So here the viceroy, not being toxic, but it does gain some advantage, some benefit by looking like a toxic species, and therefore predators tend to avoid it. Okay, another example, or another type of mimicry is known as Mullerian mimicry. This is where you have 2 unpalatable species that resemble one another. And the idea here is that of amplification. So you have, let's say, a predatory species of bird that feeds on a butterfly, and that butterfly, while being toxic itself, also resembles another toxic species. In this case, what happens is sort of a feedback mechanism where the predators start to associate anything that even remotely resembles one toxic species with other toxic species. And so they just avoid the whole lot of them. So here you have, for example, these guys look very similar to each other. These guys look pretty similar. These guys look pretty similar, not only to each other, but also to these guys here. So it sets up confusion. It sets up doubt in the predator's mind because all these species are toxic. And so it's best just to avoid anything at all that looks like them. The predators learn much more quickly as a result of this, and that means that the prey species, these butterflies, gain protection much more quickly. That's Mullerian. Two unpalatable species resemble one another. All right. Our second pattern of evolution here is that rates of character evolution differ. Rates of character evolution differ. In other words, not all characters evolve at the same rate. So this is number 2, right? Pattern number 2. So this can result in what's called mosaic evolution, which you see examples of here. The evolution of characters at different rates within a lineage. So take a look at these two adult sea urchins. So they belong to the same genus. This is Heliosidaris tuberculata. This is Heliosidaris erythrogramma. So they belong to the same genus. They're very similar, very closely related in that sense. Belong to the same clade, in other words. And here you see on the right, their larval forms. So Heliosidaris erythrogramma has what's called direct development, where the larvae develops into the adult without going through metamorphosis. So the larvae actually does resemble the adult in many ways. However, in Heliosidaris tuberculata, the larval stage actually goes through metamorphosis. It has indirect development, so it metamorphoses into the adult. So the larval stage, the larval developmental process here in this species has evolved much faster than it has in this species. And so what you end up with is larval stages that look very, very different and go through very different processes to develop into the adult, whereas the adults are actually very, very similar to one another. So mosaic evolution, the larval developmental process here has evolved much more rapidly in this species than it has in this one, and so the larvae do look much different. All right, so another example of this, body size in mammals. That is something about mammals that has evolved quite quickly. So what we see today is a wide disparity in body size in modern mammals. Compare a giraffe or an elephant, or most extreme, a blue whale to a shrew, which might weigh 4 or 5 grams. And then you have everything in between. That's a really wide disparity in body size in one group of animals. You don't see that kind of disparity so much in amphibians, for example, and you don't really see it too much in terms of reptiles. So it's just something about mammals that's happened. Mammal body size has evolved very, very rapidly. In contrast, mammals have largely retained the ancestral pentadactyly or 5-digit limb. That's a very conservative character. So body size is a derived character, but evolving much more rapidly, whereas the number of digits, pentadactyly condition, has evolved very, very slowly. That's a conservative character or a conserved character. All right, so mosaic evolution then describes how species evolve through often independent changes in individual features. Overall, an organism evolves over very long periods of time as a mosaic of the evolution of its individual features. So features do not always evolve together. They also do not always evolve independently. It just depends on the feature you're talking about. There are some features that work together, and thus a change in one causes a resulting change in the other. So these features will actually evolve together, but not all features do that. The point is that Organisms are mosaics, right? Mosaic evolution results in species that are mosaics of both ancestral and derived characters. Some vertebrates, for example, have more derived character states than humans. Think about a bird's wing. That is a much newer, much more derived feature in terms of the forelimb than the ancestral forelimb structure that humans have, along with most other organisms, most other vertebrate animals, especially mammals. Humans then have other more derived character states than other vertebrates as well. Think about the human brain. That's certainly much more developed than what you see in most other mammals. So different features evolve at different rates, and organisms are mosaics of both ancestral and derived characters. And this is why it's not correct to consider some species as more advanced, quote, than others, right? We all sort of have evolutionary baggage. All right, pattern number 3. Evolution is often gradual. So this is pattern number 3, evolutionary pattern number 3. Evolution is often gradual. Whether evolution is gradual or abrupt is actually heavily debated today. Darwin said evolution occurs via small successive changes, what he called gradualism, rather than by large rapid changes, what we today call saltation. Saltation means to jump. So evolution through saltation is referring to evolutionary change that happens in rapid spurts that then go on for a long period of time unchanged, and another rapid spurt of change with more sort of stasis beyond that. So saltation then, there are good examples of features that evolve through saltation, but there are also good examples of features that evolve in a gradualist or a gradualistic way through gradualism. And gradualism is what Darwin focused on because he saw lots of evidence of that. Intermediate forms between higher taxa like phyla and orders are often actually lacking when you look at it, and this suggests saltation is sort of maybe a primary mechanism there. But we also have some intermediate forms that are well known as well, and that suggests gradualism. If you look at the intermediate, if you look at the beak structures of sandpipers, for example, like you see here in this figure, the intermediate length and curvature bills present between extreme forms in these birds suggest small changes. If you compared this guy to this guy and didn't know anything about these in between, you might, and you realize they were related, you might think saltation. But we also have these intermediate forms, closely related species in the same clade, where you see this sort of gradual lengthening or shortening, depending on how you want to look at it, of the bill. We see the same thing here, for example, and we see the same thing here, where you have downward or curving bills. So these are good examples of where we do have evidence of gradualism, but in other cases we don't have good evidence of gradualism. Maybe it looks more like saltation, but you have to wonder if we're thinking saltation because maybe we just haven't found all these guys in between, or they're not recognized as being members of this clade or whatever. All right. Number 4, so pattern number 4 here. Change in form is often correlated with the change in function. So a change in the form of a structure or feature is often correlated with a change in that structure or feature's function. As functions change, form changes too. A good example of this, the stinger of female wasps and bees is actually a modified ovipositor, an egg-laying organ. So think about a beehive. So lots of male and female bees in that hive. The females, only one of them, the queen, actually lays eggs, right? All the rest of the females are workers and that sort of thing. So like the queen, they have an ovipositor, but their ovipositor has been modified into a stinger. The males, being males and have never laid eggs, don't have an ovipositor, therefore they never had a feature that could evolve into a stinger. Whenever you're stung by a bee, you're stung by a female bee. Males don't sting because they never had a structure that was sort of pre-adapted to become a stinger. And that's why only females of order Hymenoptera, the bees, the ants, and the wasps, possess a structure that could have evolved into a stinger. That's why only the females actually sting. So, change in form is often correlated with a change in function. All right, pattern number 5. Similarity decreases throughout what's called ontogeny. All right, ontogeny is development. So think about embryological development. That's what ontogeny is, right? This brings us to sort of a somewhat archaic concept that still has some relevance today, known as von Baer's law. Von Baer's law, okay? Here's what this says. Von Baer's law says that features common to members of a higher taxon, like a phylum, for example, often appear earlier in development, ontogeny, than defining features of lower level taxa, like orders, families, or genera, for example. All right, what this means is that species, very, very different species, like different types of vertebrates, fish, amphibians, reptiles, mammals, let's say, are typically more similar as embryos than as adults. As embryos, they share many physical features in common, but as ontogeny or development progresses and they move toward becoming adults, those similarities sort of disappear. So, again, von Baer's law says features common to members of a higher taxon, like a phylum, often appear earlier in development than defining features of lower level taxa. So when you look at all the vertebrate animals, or let's say the chordates, those that have a spinal cord, early in embryological development, if you're comparing different embryos from fish, amphibians, to reptiles to mammals at maybe the same stage, really early in development, they all look very similar. But they all have features common to the chordates, or that are characteristic of chordates. But as development progresses, and you get further in the developmental sequence toward birth or hatching, and then finally development into or toward adulthood or into adulthood, you start to see distinguishing features of their specific orders, classes, families, genera, for example, that show up, and they start to look more different. Here's a great example of that. So here you're seeing the different stages in the embryological development of humans, possum, chicken, and salamander. So we have 2 primates, we have a bird, and an amphibian. Look early in development. They all look very similar. And actually, if we had a fish over here, you'd see they all resemble the fish, because we know that modern terrestrial all modern vertebrates evolved from fish. Fish were some of the earliest vertebrates that we have on the planet. Now, later in development, we start to see unique features of the mammals, of the birds, of the salamanders, the amphibians starting to show up. And by later stages of development, now they all look very different. All right, but you can see how closely the amphibian resembles a fish. That's because amphibians evolved directly from fish. They were one of the first groups of vertebrates to evolve off of the fish line, the fish lineage. So this is an example of von Baer's law. And von Baer famously said, von Baer's law led Ernst Haeckel, a very famous 19th century biologist, to famously suppose that ontogeny recapitulates phylogeny. So Haeckel said this. Ernst Haeckel. All right. Well, what does this mean? Ontogeny recapitulates phylogeny. Basically, it says that he was saying that an individual replays its evolutionary history during its development. So if you're looking at mammals, for example, right, you're looking at mammal development, ontological development. as that mammal develops from an early embryo to a fetus and finally to an adult, you should be able to see it progress through the evolutionary stages of the mammals, right? Of actually all the vertebrate animals, going from fish, which came first, then to amphibians, then to reptiles, and then to mammals and birds. Well, that's what ontogeny recapitulates phylogeny was saying. Today, that's been largely discredited. And one of the primary reasons is because certain features, like, for example, pharyngeal gill slits, like what you can see right here, for example, that all humans and every other vertebrate has, or chordate has early in development, things like pharyngeal gill slits and branchial arches of mammals, birds, and reptiles never actually develop into full-fledged gills as embryos. They do in fish, and these features are present in all these other groups, but only in fish they become gills. In these other terrestrial species, they don't become gills, but the rudiments of them are there early on. They actually develop into other features entirely in the fully formed animal. So this idea of ontogeny recapitulating phylogeny, while it sounds good, it isn't really borne out in most cases, although there is some nugget of truth to it. All right. I think I'm going to go ahead and stop there. We've been going for about 35 minutes or so, so I'm going to go ahead and stop there, and we'll pick up with a third video lecture for this with our sixth pattern of evolution, which is known as heterochrony. But we'll get to that in the third video lecture.